Okay, hello everyone. Thank you very much for coming. My name is John Randell. I lead the Executive Office for Human Cell Atlas. As you heard in the morning talks, the Executive Office has a pretty central role to play in terms of coordinating the data flow into HCA and then out of HCA to data requesters. So I am pretty active in that process, as is my colleague Lucia Robeson, who's there in the front row. And you also, of course, heard quite a bit from Chloe Villani this morning as well, who I'll just remind you is not only a member of the organizing committee and the executive committee for HCA, but is also, among many other roles, a co-chair of what we call the Data Ecosystem Oversight Group that really helps to set policy and practice for what we call the HCA data ecosystem. So we have a few questions we want to discuss with you today, but what we really want to hear about is what the barriers are in this region to both submitting data to HCA and also to retrieving data or to accessing data from HCA as well. And so we have three discussion questions for this session. The first is really, are there any policies or regulations or technical difficulties that make it difficult to share your data with HCA and upload your data? We'll talk a little bit about what that entails. And when I talk about data, I'm really talking about sequence data, FASTQs, for example, gene expression data, and then metadata, which can include data, as you heard this morning, that could be personally identifying. So that could be, it could be medical records. It could also be ethnicity metadata. It could even just be things like age, location, and so forth. So that's the first question. It's really just about contributing data to HCA. The second question then is on the other end of the data flow, which is downloading data from the HCA data repository. And you'll note that I've mentioned here both the HCA data repository and Chan-Zuckerberg cell by gene. And that's because, as you heard this morning, Chan-Zuckerberg cell by gene is what we call the matrix store. It's the place where we store gene expression matrices so they can be visualized on that platform and accessed. So really, the HCA data repository, I will remind you, is for really for raw sequence data and also for managed access data, whether that's sequence data or metadata. And then Chan-Zuckerberg cell by gene is for matrices. And then finally, we've heard a lot about the particular challenges associated with China, again, both ways, both contributing data to HCA and accessing data. So the third question is really specifically about China. I know that, I mean, HCA, of course, has servers primarily in the United States, and that could be an issue, but we've also heard about challenges even within the region, sharing data within countries, not just including China, but also including China. And so that's for the third question. We really want to hear about sort of specific cases where data transfer has been difficult. I will remind you, in addition to where data is stored, I want to also mention that you heard a lot about sort of the technical framework for the HCA data ecosystem this morning. There's also a legal framework, and all you really need to know about that is that HCA is sort of a data custodian. We have a legal entity, which is the data custodian for HCA data. And if you contribute data to HCA, you actually sign an agreement with HCA that stipulates whether your data can be open access or if it needs to be under access controls, in which case access is controlled by the HCA Data Access Committee. And also stipulates things like what are the downstream restrictions, and you also certify, for example, that you have the proper consents and that you have the right to share the data with HCA. So that legal agreement is really critical because it's really what tells HCA how to handle your data. And so that's a really important thing that our data ingestion team, our data wranglers, will talk through with you when you go to contribute data. The other thing I want to mention is on the question 2 on the data access side, because I think there's sometimes a misperception that HCA data is not accessible in every region of the world. So I looked up the data before this session, and in the last 2 months, about 17% of our download requests came from the Asia Pacific region. And that's actually not counting Australia, New Zealand. That's Asia Pacific. If you add Australia, New Zealand, it actually goes up to 40%, largely because of one study in Australia. And about 10% of the overall data requests come from China. So as far as we know, we haven't heard any reports of sort of country-specific barriers to accessing HCA data. But if you know of cases like that, this session is the time when we want to hear about those and figure out what we can do to get around them. So I think I want to stop there. And before we really get into the questions, just ask if there are any general questions that you have about contributing data to HCA or accessing data from HCA. so that we make sure that we have a good baseline understanding before we go into the questions. I know we have an online audience too, so I want to invite anybody online to either unmute yourself and ask a question, or to just type it into the Q&A, and someone here in the room will read it out.
Hi, I just wanted to know how do you deal with the duplicate data? Or let's say I want to collect the data from of heart cells for one particular study. So I also have the raw fast queues along with the data from HCA. And which one do you prefer? Okay, I think I'm not so clear.
You're asking about different.
I mean, like, let's say regarding even the cell annotation, what you have mentioned in the morning, how does the duplicate data play a role in human cell atlas? Is there any duplicate data or no?
Jimena says you've been through the process. So.
That question is a good one, and it's not that simple. And so, so first of all, there's no data duplication between what is in CAP and what is in the metric store. And in both cases, actually, we're using the same files. It's actually coordinated. That is why there's 3 pillars of the data ecosystem, so that there's no file duplication. That is, you submit your matrices in a specific file format with the technical metadata that gets a unique universal identifier, or UUID, that then CAP can use to pull that information, so that you can also have access to the technical tier 1 metadata to help with your annotation. The gene expression matrix, what is tier 1 metadata, and then annotation metadata, is also ultimately linked to the raw FASTQ files that would be in the HCA data collection. So all of these behind the scenes are connected. Now, of course, if this has been published in other instances, so if these are published data sets and they happen to not be in the same file format, and they were deposited, for example, I'm going to use North American examples. It's deposited in the gene expression omnibus as a matrix, and the same group later on wants to contribute the remix atlas, and it's not the same file format. That is correct. That in principle it could be seen as a duplication of matrix, but we tie it with a HG specific identifier, and the main distinction is that it's in a universal HCA data format that we requested. There's another case study or use case where you could have some forms of data duplication that would be in the context of people having submitted their data to HCA in their right file format. But when we start doing data integration, you gave me the example of heart cells. And so let's say I had 12 different heart data sets, and they happen to not have been aligned to the same reference genome, and we had to reprocess all the data. Because if it turns out that, of course, if it's not processed using the same reference genome, and you try to integrate the data, you're going to have significant batch effect and artifact. So we came to realize that in certain cases, we're better off realigning everything. And so if we realign, of course, some of the values in your gene expression matrix may change, in which case it would look like a data duplication. But again, everything would be very clearly identified that this is in an HCA file format, and this is this data set that is linked to this original study, but we had to kind of realign in the scope of data integration. So this is a lengthy answer because it was actually a really good question. And it's a lot of work behind the scene to make sure that we keep tracks of all these identifiers and rationale for what things would have happened. Now, I think you're hinting at the fact that some of these are already published studies and there are already data sets out there. And so where's the duplication? Yes, there's a forms of duplication in that if it's not in the standardized file format, it becomes impossible to integrate data across different studies. And so that's why we're trying to build that HCA compendium. I hope with all these different answers, I somehow answered your question. Did I?
Thank you.
Yeah. And so this is your time. I mean, Johnny and I are here to answer questions. And so we can go question by question if it's easier. Thank you.
Yeah. So I think the situation for single-cell RNA-seq sequence with Illumina is quite clear. For other technologies like multiome, I had a quick look on the data repository, and I think there's one multiome study, but it only has the RNA data. So in these cases, how do you handle the other sorts of data for ATAC-seq, or even we were just discussing long-read single-cell RNA-seq from nanopore sequencing, right?
So I know, for example, the Retina Atlas has an ATAC-seq atlas. They actually have multiple. They have a single cell atlas, a single nucleus atlas, because they weren't really able to integrate the cell and nuclear data. So they have two separate atlases for there, and then they have a ATAC-seq atlas, and they actually put the ATAC-seq atlas on the UCSC cell browser. And then what we do is from the data portal, we're able to link out to those other resources. So we can link out to the ATAC-seq data wherever it is in the world, honestly. But the real point is that for the count matrices, those have to be on Cell-by-Gene. But we're flexible in terms of other types of data. Do you want to add to that?
That's a good question in terms of future proofing. So you're right that right now on the data portal, it is primarily RNA, and that's because it's the data set. The type of modalities is the most abundant out there. So we spend a lot of time harmonizing file format and metadata in the RNA space. Also because in the first iteration of the atlas is mostly in transcriptome space. And so that's why you see a lot of that. And that's why you see a lot of our file format requirements are in transcriptional space. To John's point, only Retina had a data set ready to first submission. That doesn't mean that we're not going to take other modalities. On the contrary, that's part of the next iteration. For next version of Atlas, we actually require that people either increase diversity, increase the breadth of modalities that would bring us to multiome. And so as we work out the details, we will come up also with standardized more file formats so that we can also integrate multiome data. It became very clear within the last 2 years of trying to do data integration in the RNA space that even though that seems obvious, there were over a dozen different file formats when we started to do the work. And you're smiling, you can imagine the nightmare of trying to integrate 12 different file formats. And so I think we should learn from these mistakes and not do the same for our attacks. So this is at a point where probably at some point in 2025, we'll do a call and ask who has opinions and thoughts, because it was not just a few of us coming up with these recommendations. It was through a fair amount of outreach and socialization. So if you have specific thoughts, I'd love to hear them separately or now, so we can help. For now, for sake of making sure, and also the matrix store from Cheng Zuckerberg was not set up to host them. This is what John was hinting at. So that's why as a temporary solution, it was put on the UC browser. The HCA data portal serves as a front end so that you can find any data sets going through the same front end. So the HCA data portal is supposed to be your source of ground truth for HCA data sets. And from that front end, you can be reoriented to the right data sets. So there's like two parts to your question, which is like, is there going to be, I guess I'm reading between the lines, but are there going to be other data sets than RNA? The answer is yes. Are there going to be standardized file format beyond RNA? The answer is yes. Is it ready now? The answer is no. Why is it not ready yet for ATAC-seq? Is because there's not enough data sets out there that have been submitted, so it becomes very hard to kind of develop these standards.
Yeah, it's a good point, and good to hear that it's at least being thought about and planned. I guess there's probably, I would say, a lot of data that's out there and maybe hasn't been submitted because the infrastructure maybe not there to handle taking in the data as well.
Yes, I guess that's part of it, but I will say, I'd love to hear from you and others where you've seen a lot of that data, because when I browse the literature, there are some data sets, but there's not an abundance of that data sets just yet. And that's generated in a slightly standardized way.
Okay.
But are you talking about multiome or ATAC?
Both, yeah.
Oh, yeah.
I think the one thing I want to add is that the HCA data portal itself can actually accept any file type. So in theory, we can accept multi-ohm data as well. The caveat though is that we're really focused, particularly for other omics, we would probably only do that for data sets that were actually actively being integrated into atlases. So it's not just like we can't just sort of accept data from anywhere. I mean, we could theoretically, but we just have to prioritize that. But in theory, there's actually not, I mean, I think there may even be some spatial data actually already. But so it's not so much a technical barrier, I think, for us, if that makes sense. I did want to show, so we got connected to the internet, so I can show you, this is the HCA data portal. You saw a bit of this before, but it's useful to have this live because I can show you a couple of things that we were just referring to. I want to first actually go to the atlases down here. I'm so used to Macs, but this is the iNetwork homepage or iNetwork network page on the HCA data portal. And down here is what is the first atlas that's come from the iBio network. This is actually specifically a retina atlas. They have other atlases that are in the works, optic nerve and so forth. And if you click on that, then what I was referring to before, you can actually see there all of the component atlases that make up this retina atlas, the first published atlas from the iBio network. And so if you scan down this list, then you can see that we link to cell by gene for most of these because we're linking to expression data. But as I was just talking about at the bottom, the ATAC-seq atlas is actually on Cell Browser. And so you can actually get the ATAC-seq data off of Cell Browser. So that's what I was referring to there. And then I want to go back. Oops, went too far. Yeah. Oh, thanks. I was actually going to go back to the website. So this button right here called Explore Data. This is what takes you to what we call the Data Explorer. And this is actually where you look for individual source data sets. So the Atlas pages include lists of all the source data sets, but you can actually explore all of the data sets in the data browser and the data repository here through the Explorer. We talked a little bit about managed access. So this is a record for one of the managed access data sets. You heard about this morning, that in the upper right, you can request access to that data set, but anything with a green button, you don't need to request access to. It says that access is already granted. And so you can click on this and you can see background information, the contacts for a data set and so forth. And you can also then get the links to download and also export the data sets either locally or to other services, particularly the Broad Terra repository. So that's basically a really quick look at actually where to get these data sets, but I want to make sure that people were aware of that. Let me refresh this. No, it's not refreshed. Oh, this is the PowerPoint, isn't it? Okay, yeah. I actually wanted to go back to the live website there if I can. Yeah, right here, actually. Capture this link. Yeah, but we actually added to it, so I want to add, yeah. So I was going to go back to the browser again. Oh, we actually already have it up. Okay, good. And then, so I also just wanted to remind you, we had trouble getting this. Yeah, we had trouble getting this graphic up before, but just a graphic. I'm a visual learner, so I have to see things. I can't just listen to them. So this is what a sort of another representation of what you heard before. So this is just a reminder of what we were just talking about, that the matrices go on cell by gene, the capacities go in the data repository along with managed access metadata in particular, or biological metadata. And then the cell annotation metadata we heard goes to CAP. But what Chloe was just referring to is really important. We try to make this as painless as possible for you as a data contributor. And so there's a team behind the scenes that will help direct the different components of your data to these different resources. And then of course, you heard about the integration team in the morning that actually then takes everything, puts it back together and starts integrating the data to make atlases. And then here's a diagram that shows what I was talking about the legal flow. And I think this is actually really important because this is actually more than the technical aspects. This is where a lot of the barriers, or at least perceived barriers, can come to when we're talking about data contribution. So I just want to take you through this really quickly. I mentioned that HCA has a nonprofit organization that serves as what we call the data controller, the custodian of data legally for HCA. So on the left, you have the data contributor, and actually not just the individual PI, but also their institution that has to co-sign the data contribution agreement to HCA. That's the blue arrow in the upper left is that data contributor agreement. What you haven't heard about, but you don't really need to know, but what you haven't heard about, thanks. What you haven't heard about is the three institutions that work behind the scenes to operate the data repository. You don't really need to know this, but just so you know, principally speaking, the data wrangler team is based at EBI in the UK. The data repository is operated by the Broad Institute of the United States, and then the data is indexed and served to the data portal by UCSC. And so all of that is behind the scenes. So if you're a data user on the right, you and your institution sign a data access request, and that then is then funneled through the data portal to these behind the scenes actors that then route the data accordingly. And in the bottom right, you can see the data access committee. There is one data access committee that controls access to all managed access HCA data. So if it's managed access data, it follows that green arrow. Your request gets routed to the data access committee, and the minute that you get approved by the data access committee, you then have access to the data portal to that data set. That orange button will become green for raw data. Yeah, this is for raw data or managed access metadata as well, right? So this is all basically the data repository, the FASTQs. So the cell by gene is a different thing. You don't need to worry about it. But the point that I want you to really take from this is that there isn't a legal agreement at both ends of the data flow. So the contributor signs a legal agreement, and then the user signs a legal agreement, and the user's legal agreement is connected to the terms in the contributor's legal agreement. And that's how we assure that the users of your data are bound to the original terms and your original understanding of how your data will be used by HCA members. And that's essentially not just HCA members, anybody. And so that's really how we assure compliance with whatever your local regulations are. So to give you an example, for example, we've been piloting a number of data sets for managed access. We have 2 data sets coming in from Japan, one from Thailand. Those are all from the IATA project, and then one from South Africa. And so an example of the types of issues that we've had to work through in this process are sort of national regulations, like Genome Thailand, I believe, is very concerned about commercial use of data. And so as we've talked through the data contribution process, we've had to sort of assure them that we can protect their data in accordance with their national regulations. And those are the types of issues that really relate to this first question. We're really interested in hearing even what your concerns might be if you were going to contribute data to HCA. What do you think might be the barriers, legal, technical, institutional, otherwise, that might prevent you from contributing data to HCA?
And I mean, we had questions so far about like using the data, but just kind of want to add, why are we asking about this for a data set to be considered for Atlas assembly to be integrated, right? And then you heard about data integration. I just want to do a side note. The team that integrates doesn't necessarily do the downstream queries. It's just to kind of how to do all the heavy lifting for the data integration so that there can be an object that all of you can use for your favorite queries. But back to, if you're contributors and you want to contribute a data set to an HCA, we can only integrate your data sets if you contribute the matrix, the gene expression matrix with the technical metadata, as well as we know where your raw data is. And that's because, and that was one of the first questions I was asked, it's tied to the first question I was asked, if the data was not aligned the same way, we need to be able to potentially realign. We need to future-proof these atlas we're building because, you know, they will evolve over time. We're going to have new computational frameworks. We may want to look at genetics. Who knows, depending on what your RB allows. So we need to know where these raw FASTQ files are. And ideally, if they're part of an atlas, first version of an atlas, they should all be compiled together. And so otherwise that sets some boundaries in terms of which data sets we can integrate. And so in many cases, when you publish, the journal will ask you to deposit your data somewhere and to, you know, reference where they are. So we would like to know what would be some limitations for submitting your data, or it could be anything else you'd like to raise. But we've heard a lot of things over the last months and year about some challenges in Asia, and based on the statistics we're reviewing, it's not clear to us where the barriers are. And so this is a chance to tell us about it and find solutions together, or anything else you'd like to ask us.
And again, if there's anybody online that has a question or a concern that they want to raise, please do. But yes, we have another question here in the room.
Just to start some more discussion. I think there was a paper recently from Gamze Gerso who says that you can identify individuals from the count matrix. So how do you think about these problems also?
So HCA's policy is, and this is really this is exactly the reason for the tier 1, tier 2 metadata schema. And so what's associated with the count matrix is just the tier 1 metadata, what we think of as technical metadata. That's all sample preparation, project information on the project, what technologies you're using. There are like 3 fields, I think, that.
Yeah. So
Oh, the actual data. Yeah. Sorry. Yes. Right.
right, there's an argument that the count matrix is personally identifiable information.
Right.
And so then it should be under some protected access.
Yeah, I did actually see that paper. We have yet to have a conversation about that paper. Actually, that's probably something we should raise in the data ecosystem group. For HCA itself, now HCA doesn't actually hold your count matrix, so this is really an issue for cell by gene, but it's an issue for us if we're actually like linking out to it, right? And so it's also a discussion that we'll probably have to have with their team.
Somebody else's problem.
It is our problem because it's, you know, we are the custodian of the data, but it's something we'll have to work out with our partners. Yeah, but yeah, that was a striking paper, and to be honest, it's actually something I've been worried about for a while because we all know how little information you actually need in order to identify an individual, whether you're talking about street records or genetic data or whatever. So that's a really good question.
That is a very good question that obviously goes beyond HCA. Actually, it ties into all of us with RB protocols and consent form, because, you know, at the end of the day, it's not just HCA that's responsible for the data. It's every one of our institution that has approved our IRB, where we commit to the patient of having some de-identified, you know, data set being submitted in some open data repository. And so that question is like, is it now such that we can no longer do this? So it's an important legal question, but it goes back to the consent form and how much information do you actually need to identify someone? And so this connects actually to how John started answering the question, which is, of course, like on the matrices on his own, definitely is like a footprint, definitely could identify you, but can really identify you if we start connecting it with additional information to really hone in onto you. And so we spend a great amount of time thinking about what you've heard me and others call tier 1 metadata to make sure that it's absolutely impossible to use this to identify you. And so really focusing on what type of metadata does one need to do data account for batch correction. So if you tell me it's cell or nuclide, I will not identify you. But the only identifiers would be in this terms of broad demographics. What do I mean by broad demographics? Male, female, because you could have some gender imbalance. And when you do data integration, that's not going to identify you. Age could identify you. So we made these age brackets very large. So that's actually very hard to identify you. We specifically moved away from identifying disease or cause of death because that can actually identify you. We do try to understand if the samples came from a living person or someone that passed away, because that can actually be tied to technical artifacts in terms of number of genes and mitochondrial signature. But we don't want to know if it's necessarily, and Lucia, correct me if I'm wrong, but like if it's like suicide, car accident, we don't want to know that information in the tier 1 metadata because that could identify you. We don't want to know about details of a disease because especially if it's a rare disease, that will identify you. So we were extremely careful in thinking about, what information do we need out there publicly to help this data integration, and making sure that none of them could in combination lead to identifying you. It's not to say that other consortium don't do it outside HCA, but within our HCA policies, we were very thoughtful about this so that maybe it's true that your matrix is a special footprint, but alone without a bigger data set, a bigger repository that connects different pieces, it will be really hard to figure out that today it's me. And so, yeah.
And I'm sort of thinking out loud here, so I apologize. I'm thinking through my answer as I'm saying it, but I think we have a little bit of protection in HCA because unusually, actually, HCA's policies in terms of what information we protect and make open are actually emphasize protecting the metadata. In fact, we actually, in our formal policies, including our sort of regulatory documents, we actually assume that all of the FASTQs will actually be open access. meaning that we start with the assumption that not just the matrices, but the sequence data will be open access. And then the question becomes, how much of the metadata do we need to protect? So we're much more conservative than many organizations and consortia are. And the age bucket is a good one. So there are a lot of repositories where you can actually find specific ages, and that's not HCA's policy. We would bin ages across like decadal or at least 5-year, but preferably 10-year bins. So in a sense, we've sort of anticipated the problem with the Cal matrices because it shouldn't be a problem because the really sensitive metadata is still under lock and key. That's kind of a long-winded answer, but it's not that it's not a concern. It's absolutely a concern. But like I said, we still need to work through the implications.
Perhaps to kind of clarify, historically, when we set up HCA, or I guess for people online, we should be on the mic. Historically, when we set up HCA, and perhaps naively, we wanted everything to be open access, including fast queues. And it became very clear that, especially with samples being collected in medical institutions, that would not even be an option. And so things have evolved over time. And so we've, like to John's point, we've been, extra careful. So this was just a small nuance to explain why we had assumed we could do everything open access, but it's a really good question. Yeah.
It does actually bring me to another interesting example of what we're talking about in question 1, though, which is what we are already running into just in the early days of taking in managed access data. Is often we run into a situation where the contributor's assumption is that the metadata actually, to a quite surprising extent, will be open access, and it's just the sequence data that needs to be protected. And it's actually the reverse in the HCA policy. And so what we try to do as we ingest the data is basically use the highest level of protection that would be mandated by either HCA policy or by the contributor's policy, if that makes sense. But what the risk there is that you end up putting more data under access controls than you might otherwise want to. So that's still a balance that we're trying to find. But it's a really excellent question. Are there any other questions pertaining or concerns pertaining to your ability to contribute data to HCA?
Can I sort of frame this slightly as a question to you about how much of the responsibilities on the funders and the people funding data collection, and how much of that is baked into the grants and that come through this? And I say this as I run GigaScience. As a journal editor, I see lots of I peer review publications with lots of data. There's the big issues, number 3, Ministry of Science and Technology in China doesn't let data go out of China. But even in China, and as you've seen from your own metrics, people can't access their own data inside China because a few small labs kind of see this as their own private walled garden, that they just get publications out of this data, and they're not really been held to account by the funders to share this, right? So but the people funding data production here, how strict are they on if we're going to give you money for data production here, you have to share this, we're not going to give you any more grants if you don't, right?
Yes, you can speak to this, I'm sure, as well. I know it is a requirement. For example, I believe people in this room would know better than I would, but I believe CZI, for example, requires that data be uploaded to Cell by Gene and actually also to the HCA data repository, and then they fund a service that facilitates that. And I know this question came up in the AIDA meeting yesterday because that does make it difficult to fund projects in China.
But in Asia as a whole, like how much of the data production is funded by CZI, and how much is from like local funders who may not hold people to account in the same way?
I don't think I know the answer to that. As far as I know, I haven't run into any issues with this myself, but as I'm not a data generator, so I can't really speak from that perspective. I don't know, Chloe, if you have an idea.
It's a good question, and I'll take a detour to answer your question. In that, unlike other big consortium where it's top down, you have a pot of money with some funders and it trickles down. We're a grassroots initiative, which means anybody can contribute their data, which means it could be that it's from one. I mean, we do not get too much in the weeds. We have a funders collective where we can help set up priorities for HCA, and funders can listen to us and then go on and create their own research funding opportunities across different countries and regions. And then they have their own policies. Because we're a grassroots initiative, sometimes it comes from one of these mainstreams of, RFAs that came through our funders collective, or it could be like, I'm going to use myself as an example. I got funding that has nothing to do from these funders collective, but I want to contribute to the bigger mission of HCA. And so I came with my own pot of money, and it's actually true in this case, and generate data and contribute it. And so if we were one of these consortium that we were very top down with a pot of money, then it'd be very it would be easier to enforce this, to say, well, your HC has this pot of money, we're giving it to you, you must bring it back. But that is not how HC works, being a grassroots initiative. Now, some of our main founders, John mentioned CZI, Hemsley, for the God Atlas, they definitely is part of the requirement. Also, if you want actually to contribute to HCA, if you want to be an HCA member and be one of the data contributors and contribute to Atlas and then benefit from some of the resource being benefiting from some of our infrastructure that is either like data management, data access, but also analysis, contributing to Atlas assembly beyond your own academic paper. You were saying like your own little garden protecting. So you publish your own paper, but you want to elevate your work and get a second paper out of it. And the second paper would be through this Atlas consortium. Then you must deposit your data. That's part of the ground rules to be part of HCA. And so if you cannot deposit the data, then there's no way for you to be part of it. So it's a good question you have. And there's pros and cons in being a grassroots initiative. I think one of the many benefits is that everybody's welcome. There's no barriers, right? It's way more democratic. It also makes a better scientific product over time because it can iterate and encompass a greater breadth of scientific expertise. But some of the downside is that we don't always have the stick in terms of the fundings. But if you want to be part of it, that's kind of the ticket to be part of it.
That's the carrot, yeah.
That's the carrot, yeah. And then with time, maybe the carrot will become a bit more obvious. So for the publications, for example, especially as we iterate atlases, it's very important for us that these atlases are reproducible, which requires transparency, not only where the data is, but where the code is and how did you, get this product. And so it is part of our requirement that there's a data availability statement where the data is. And we understand that for version 1, because a lot of these infrastructures and rule came about as we were doing it, that some people will have to be grandfathered. And so perhaps some of your raw data will not be in our repository, but you still need to have your raw data somewhere and you still need to tell us where it is, and it still need to be accessible to some forms of committee. Otherwise, we cannot take on your data. That's a very good question.
There is actually a very direct example of what you're asking about, which is Europe, because the European Commission funded 6 consortia at the very onset of HCA, and they required that the sequence data be uploaded to HCA, and we actually had no way of complying with GDPR, so we actually couldn't do it. So a lot of our managed access system was actually driven by that, and is built to be compliant with the EU GDPR regulation. So we're actually now able to accept that data, but it took several years to actually set that up. I think I'm most interested in moving on to the third question. I think we only have about 15 minutes left. But I guess first on question 2, has anybody actually encountered any difficulties downloading data from HCA? I hope the answer is no, because as I said, I have not seen any evidence of this. I will take this opportunity to say that if anybody requests a large amount of data from HCA, we do have limits on on the frequency of requests. So you may, if you want to download hundreds of data sets, you might run into that issue. But in that case, you just contact the executive office and we can make sure that you don't get blocked. That's right. I should actually say that. We haven't said that. HCA does pay for all egress. So your downloads are free. And in order to preserve that, we need to make sure that that's really the reason we want to make sure that, we don't have AI bots just scraping data repeatedly and racking up large charges, which would mean we have to shut the entire system down. So, but yes, all storage and all egress is free of HCA data. So you don't have to pay for any of this. That's true. That's true. Yeah. And I should say that that's also thanks to the generosity of CZI, which helps us cover those costs. Okay, then on question 3, I guess I want to preface this by saying, first of all, that we have plenty of researchers in China who are HCA members. It's about 5% of our membership, about 170 scientists in China. So there's a lot of really fantastic research. We've heard about some of it coming out here as well. But it's a challenge of sharing those data with HCA, but then also analyzing Chinese data against other HCA data. That's also a challenge we've heard as well. But honestly, I was actually surprised to hear that there are data sharing issues even between Hong Kong and mainland China. And so my sense is that this is not just an HCA issue, but it may be even an intra-region issue as well. And so we're interested in hearing HCA specific barriers here involving data from China, but also just anything that you've experienced, those of you who are not from mainland China with that data, or those of you who are from mainland China, with data sharing, even with the countries in the region, and how HCA might actually be able to help those types of problems. So does anybody have any experience or any concerns here? Perhaps not. Yes.
I'm not a scientist. I'm just an belong to the administrative sector of the government. And probably it's not what Chinese researchers concerns, not Chinese researchers concerns, probably what the Chinese government concerns. And so it's not the matter of science. So if, for example, if we can make some agreement. So if China provides 10 times data than the data Japan provide, probably for Chinese government staff, it seems so. And Japanese researchers use that data freely. Probably for some government staff in Chinese government staff, it seems rather unfair for some people in Chinese government. And so if we can make some agreement, for example, in the futures, IP holders, which that IP was get by using HCA data, that IP holders cannot exist his IP within China against for one or few companies which Chinese government designates. So it means within the China mainland, in some under some condition, other countries' company can't use their IP. So such kind of agreement, it probably also very difficult, and some definition of which IP is the target of that agreement. But probably something like that is one of the solution, I guess.
That's what is the right agency to talk to about that?
Yeah, Ministry of Foreign Affairs,
Okay.
and in this case, probably also Ministry of some healthcare, and also Ministry of Science and Technology somehow, but mainly Ministry of Healthcare.
Okay, good. Yeah, that's good to know. I will say HCA has always been careful not to get involved in IP negotiations where we don't claim IP ourselves on anything. So that would, as you say, it would be difficult, but it's certainly worth looking into, I think. Yes, I think so. That's not actually specific about China, but yeah. Yes. Let me see if are there any other comments, particularly sort of on either?
Yeah, I know some background about this. I think this is mainly because the government released. You want to know the department? Just check this called Ministry of Science and Technology of the People's Republic of China. So they released a regulation, not a law, but a regulation regarding the human genome resources. Right? I think for human, because the government believe, this is my understanding, believe this is sensitive information, should not be, shared globally. But I think under certain circumstances, this data can be shared.
Mhm.
But you need to get the agreement from the government.
Yeah.
The process is a little bit, will take a long time, but still, once get approved, the data can be shared. But I think you need someone, if HCA want to, collect the data, you need to find someone in China, a scientist in China who can just lead this project to negotiate with the government or apply to the government. But apart from human, samples, I think other sample type like monkey or mouse, I think this type of data will not be considered sensitive, right? Can be shared. But I think you need to treat this differently. Right. Yeah.
So I'm glad you raised this, because this is actually the core question that I would like to learn more about, which is, I can understand that applies to genetic sequence data. What about gene expression data, which, notwithstanding the other questions
Yeah.
heard earlier, historically has been thought of as non-identifiable. So, or process de-identified, whatever word you want to use. Is that shareable, or is that also subject to the regulation?
To me, my understanding that FASTQ file is very hard to be shared,
Sure.
but gene expression matrix it should be okay, right? Because the government, treat the FASTQ file, genomic reads, right? The read from the not maybe genomics or transcriptome, they treat this information as sensitive because you have the ADGC information,
sure,
right?
sure.
But for the gene expression data, I don't think they care about that. And also, you want to publish paper, you can upload the FASTQ file maybe in some deposit the data in some places, right? Held by the China.
Mhm.
Some place in China, right? Upload the FASTQ file, but you have to upload the gene expression data, right? And other people can at least repeat your result, right?
Yeah.
Yeah.
I think that's where the opportunity is for us, because in theory, we can link out to any repository for FASTQs. So if this is really, I think, an awareness and sort of just a conversation that we want to make sure that people feel comfortable with the gene expression data being shareable, because if that's the case, then I think it actually opens up a lot of possibilities.
Yeah, because it will take a long time to get approval from the government to upload the FASTQ file like abroad. So some of the scientists, they don't want to waste their time to publish the paper. They just want to publish the paper as soon as possible, right? And so they will not just sell the FASTQ file, but in order to get the paper published soon. But if we want to collect the data, I think it's doable. We just need to find some key members from HCA. Maybe they can help you to find the right department or right person, right? So that maybe we can just communicate, to the government, right? The government may just agree that this is pure scientific study. Some of the non-sensitive information can be shared. At least I think non-human data can be shared, right?
Yeah. And I think we're actually starting that conversation with some Chinese scientists at this meeting as well, just in side sessions. So it's great to know. It gives us a couple of different avenues. And I think we could even talk about calling out the special case in the protocol, the ingestion protocol, and maybe having a second path for FASTQs if in restricted situations like this. So that's actually really good to know. Thank you very much.
I guess This was extremely helpful. And I guess to clarify, we were even told by other members that they could not even contribute the gene expression matrix. I mean, I completely understand the challenges tied to RAF-FASTQ files for all the reasons you mentioned and many more. But if we can share the identified matrices, that enables bringing everybody on board. And there's other solutions that say, I mentioned the example, oh, we didn't use the same reference genome. As opposed to sharing FASTQ files, there's an opportunity to have collaboration where we share a script back with colleagues in China. I said, hey, can you just reprocess your FASTQ file so we have the same reference genome? And then which we've actually done currently, not just, it's not specific to China. Actually, we've done with other colleagues around the world that couldn't share their data. So we just shared a script and everybody reprocessed locally their own FASTQ file. So we had all the same metrics, and then it became very easy to share matrices. But I got the signal from other colleagues, other members of the HCA community that they could not even share matrices. And this is where I'm like, well, I don't even know where to start if we can even share matrices. So you seem to be suggesting that there might be a door to sharing matrices. And if we can just figure out that path, then I think there's a lot of opportunity, but yeah.
Yeah, no, this is it's a really critical issue. So definitely.
And then you mentioned other species. It's not that we're not interested by other species, but as the name calls, we're being the humans at Atlas, we're a bit more focused on the human. No, but it's a good point because we actually have an organoid bionetwork, for example, and there's definitely communities that are interested in, like, for example, the brain bionetwork is analyzing the brain across multiple species, different primates, marmoset, mouse. So no, it was a relevant question. And because one wants to obviously translate what is meaningful in terms of translating between species, although it hasn't been one of our main priorities for now. Yeah.
I think we're basically at time now. We were going to raise one other issue, which is about commercial use. So I'll just say a really quick word about that. And if anybody, sorry? Maybe we might be talking about different issues. Yeah. So this is the one other issue that I've run into is that commercial use is sort of defined differently in different jurisdictions. And so we run into this where in some countries they would consider any use by a for-profit corporation to be out of bounds, to be not allowed. Whereas in other countries, there's a distinction made between basic research by a for-profit company and actual drug development or product development efforts. And so that's just another issue I think people should be aware of, that in the HCA context, when we talk about allowing commercial use, it's just a conversation we need to have. So if you have any restrictions on commercial use, basically, and you want to contribute data to HCA, just make sure that you raise it with the data wranglers when you, the data ingestion team, when you submit your data. So I think we're at time. Yes, thank you very much for participating. Your questions were fantastic. They really get at the core of what we were trying to learn about here at this meeting. And so we really appreciate it. We encourage you to reach out to the executive office if you have any questions about data contribution or data access. Thank you.